Hey everybody, I'd like to invite the speakers to turn on their cameras. I am Susan Pell, and I'm the Deputy Executive Director at the U.S. Botanic Garden. And I want to say thank you to the three speakers from today. Really fabulous talks. And this is your opportunity as audience members to just bring out all of your questions you've ever wanted to know about all of these different wonderful relationships that have just been talked about with plants and other organisms, and all of the weird and wonderful things that we've just heard about. So please go ahead and type in your questions into the Q&A box and we will address those. But I'd love to ask panelists to sort of start off as maybe what's the most surprising thing that you've discovered about plant interactions with other organisms in the systems that you've studied? So maybe we can start with Jay.
All right, I'm unmuted. Hey, I mean, there are so many surprises when you're working with parasitic plants, and you saw from the talk. I mean, there's just any amount of time you spend botanizing and doing natural history, the longer you look at something, the more interesting stuff you find. But one thing that really surprised me after studying Hydenora for so long is that it's a true parasite, and a lot of parasites are very conservative, right? Because it doesn't help to kill your host, right? And we did some studies looking at like sort of water loss, wondering how this thing abstracted water and carbon from its host. But one thing we found was that the Hydenora vegetative body loses water like a barrel cactus, meaning like it doesn't, like it's just so conservative. And occasionally we find some hosts that were dead, but it's a stressful environment out there, things just die. But I think just the sophistication of the symbiotic relationship, right? How conservative the parasite is, how slowly it grows, how little water it actually uses from its host.
Great, thanks. Naomi, would you like to tackle this one next? What's the most surprising thing you've discovered about plant interactions that you've studied?
Oh boy, that's a hard one, because I've studied a lot of different kinds of plant insect interactions, but for the ones we're talking about today, I really was astonished that the plants would respond to the smell of the ants. And I hinted at it, but really, I mean, we obsessively did this experiment over and over because it was such a surprising result. I wanted to make sure that it wasn't an artifact of some other thing that we just weren't thinking about. The last thing we did, the last caveat, we thought, oh, well, wait a minute, we're not thinking about maybe their spores coming from the ant colonies that are going into the plants and infecting the plants with the endophytes that in fact are causing this, which would be nice because it would solve our question about what the fungi might do for the plants in the system. But we did sequence them. Very fortunately, we collected them for another reason, and so we had all the material and we just sequenced them. It was there were fungi there, fungi and bacteria, but in equal amounts and right across the board with the controls too. So that's not the explanation. And we're back at the odors. Of course, I mean, it's interesting. A lot of the compounds are highly conserved from animals and plants, and you do see the compounds in the odors that look like they should be active for the plants, but that doesn't quite explain the specificity of responding differently to different species. It doesn't, and the branching. I was very astonished by how fine-tuned those interactions were. So.
Yeah, I was actually really blown away by that in your presentation. It's an amazing result that you have. I'm wondering if you see that in nature, if you have done any sort of studies looking at the branching patterns and whatnot, the form or the habit of the trees. If you can kind of
the trees growing in response to the ants differently. That almost everyone who studied the system has commented on that. But there are not many studies out there of plants responding to insect smell. So there is one of I think it's a goldenrod responding to the smell of a midge that parasitizes it. If I remember this correctly, it was a Consuelo de Moraes did a nice work on that. She had also looked at how daughter finds its host, and so sort of plant interactions. But this business of actually responding to the smell of the ant, that there are not many examples out there, but that's why I was saying it may be more common. It might be so subtle that it's hard to pick it up unless you look at a system like this one that's so very highly sort of tuned, where the partners are so well tuned to each other. So
yeah, thanks so
a
much.
lot of fun.
Absolutely. Posy, I'm going to give you that same question. What's the most sort of surprising thing you've discovered about plant interactions with your research?
Yeah, it's a good question. I think, this might seem like an obvious answer, but I continue to be surprised by these cryptic symbionts, fungal symbionts, and how, they just show no sign of their presence yet when plants are inoculated with them prior to pathogen infection. You can see these really profound consequences for the severity of disease. And it's amazing to me that, this function went undetected for so long. And it's probably because in nature, it might be, often subtle or non-existent, and it's quite variable. But yeah, I guess I'm just continuously surprised by that, these invisible symbionts can have important consequences for plant disease.
Absolutely. And you've got a follow-up question from the audience here. It says, do you envision manipulation of plant microbiome as a future replacement for pesticide application in agriculture?
Yeah, that's a fantastic question. I certainly, hope that that is a potential application of some of the work that I talked about today. There's definitely a history of, working with pathogen antagonists in agricultural systems. Biocontrols have been around for quite a long time and have been used to combat disease and, crop disease. I think, the history of biocontrol is sort of mixed. It's not always successful. It's really hard to control the abundance of a single organism. And I think, the angle that I am hoping might work a little better is to, rather than trying to control the abundance of one single, silver bullet antagonist that's going to modify or protect a plant, rather, can we control the environment to encourage the development of beneficial communities? And by controlling the environment, I think maybe the most efficient way to do that is to control the host environment via genetics. So trying to gain a better understanding of which plant traits, maybe even more specifically, which plant genes are associated with these beneficial communities. If we can figure that out, then I think there is a potential that we could select for these traits in agriculture in order to more sustainably manage disease and raise crops.
Yeah, that's great. And that was something that I had thought of too when I saw your presentation. Really, really fascinating. So I've got a couple of follow-up questions for you, Jay. So one is, you said that Hydenora is an ancient parasite. What plant was the original host?
That is a great question. And at the base of the Hydenora phylogeny, you can see there's the sister genus Prosopanchi, which parasitizes like several families of plants. So like it could be anything. And then in Hydenora itself, it's Fabaceae. But I don't want to guess, but I know how to figure it out. I mean, we just need to do whole genome sequencing of these parasites and then look for horizontal gene transfer, like it's been shown like in Rafflesia and tons of other Orbanki and lots of other things. And then just look for the signals of the ancient parasites there.
Cool. And one other one for you, let's see, is, actually it looks like, yeah, here we go. Can you talk about the relationship between the Hydenora species of Africa and South America?
Oh, so there's no Hydenora in South America. So it's the sister genus is Prosopanchi, and that is in South America, and then the old world species are all Hydenora, so Africa and Arabia, southern Arabia. And I mean, it's probably an ancient classic Gondwanan vicariance based on the dating.
Great, thanks. And then I've got also, it looks like a follow-up question for Naomi. Do you think non-ant plants respond to smells in the environment?
Oh yes. I think they may very well. It just may be harder to detect how they do that. It's hard to ask a plant to answer a question, and it's easier if the plant depends on something like ants for its defense, because you can play around with with those components and try to get an answer that way. But I think it's quite possible. It just may be a much more subtle effect, and so it's a challenge to figure that out. So.
Great, thanks. Posy, you've got a couple of more questions coming in here. So considering how slow growing endophytes are, do you suspect that the consequences to the host might change throughout the lifespan of the fungus?
Yeah, that is a good question. So, most of so the work that I talked about today, these endophytes are occurring on cottonwood leaves, which is a deciduous plant, right? So the leaves are out in the spring and summer, and then fall off the plant when the plant is dormant in the winter. And so during the season when the leaves are on the plant and it's growing, the endophytes there's time for the endophytes to colonize and to grow on the plant. But I certainly think that the phenology of the plant and the fungus are important for the actual protection that the fungus is offering. So for example, early on when the fungus is on the leaf surface and invading the plant and getting into the plant, that could be a time when direct interaction with pathogens that are also on the surface of the leaf could be important for antagonizing a pathogen and reducing the pathogen's ability to get into the plant. Once inside the plant, then the endophyte could have sort of a different set of mechanisms that underlie its benefit to the host in terms of host resistance. That could be maybe more involved in modulating the defense response via modulating host immunity. So I think that as the endophyte sort of colonizes and invades, that process will be associated with different types of mechanisms that might be involved in disease modification. These are really, the diversity of endophytes out there makes it a little bit daunting to think about all the associated mechanisms that are involved. And so, there really isn't yet a model system, I would say, for a plant horizontally transmitted endophyte where we where all these mechanisms are really well worked out. And in part, it's because of this diversity and trying to first just document all the different types of interactions that are, or the different mechanisms that are involved.
Great, thank you. There's kind of a general question here. Do any of these symbioses include algae? So this would probably be for Naomi and Posy, I think. Or Jay, you probably have just knowledge generally too about this as well. So anyone can answer.
You mean like a lichen would have algae, or a cycad, or but not acacias. I mean, not algae, I don't think. There may be lichens growing on them, but not in the ant plant system itself. That's a good question.
Yeah, I can say, like, I went to graduate school in Louisiana, and there's certainly a lot of algae that grow on plants there. So maybe, I don't know, in your system, Posy, if there's algae that they're finding as well.
Yeah, that's a good question, but not that I've sort of looked into or have a good answer for. But certainly, yeah, as Naomi said, there are plenty of lichens out here in the Pacific Northwest, especially and associated with cottonwood trees. And
absolutely, very cool. Let's see. So another one for you, Posy. This is a fantastic story. I wonder whether in the 2 natural biomes that you looked at, other species had the same or different fungal associations as the populace. And if very different, then the diversity of fungal species in the ecosystem is an amazing resource for interactions. And I think with this, we could sort of broaden this too to the other panelists and thinking about, we're looking at sort of one system or a couple of systems, and how do those interactions maybe have an impact more broadly on the other organisms, other trees, animals, et cetera, that are around those systems that we're studying. So maybe start with Posy for the specific question, and then if Jay or Naomi want to weigh in.
Yeah, that's an interesting question. One way that so I guess I'd start by saying I think a lot of the endophytes that are found in populace are also found in kind of neighboring plants. There's a lot of overlap in the communities from plant to plant. Although, of course, there also is there is host specificity and even genotypic specificity, as I showed. But one way that I think about that sometimes, or think about that question, is the degree to which plants might host pathogens of neighboring plants in their tissues, almost as a sort of a mechanism of apparent competition. A way to, to compete with the neighbor is to host the pathogens of the neighbor. And so that's one sort of hypothesis I have, because in some of these studies of cottonwood endophytes, we find all sorts of pathogens of other plants in there. And in particular, a lot of these common gardens are found in areas where there's agriculture is prevalent, and so we'll find things like grape and apple pathogens in the cottonwood leaves. And nearby we've got apples and grapes being grown in rows. And so I think I think apparent competition is one way that I think about how the microbes found in one plant might influence other plants in the community.
Awesome, thanks. Naomi, do you have thoughts about this?
Biological warfare, that's, I think that's a great idea. I was going to say, and I think I'm going off topic, but I was very surprised by the sort of bottom-up effects, and really this has come largely out of reading and thinking hard about papers that Mike Kaspari's been writing about the importance of sodium for ants. But we were astonished when we looked at the soil composition around these ant plants that the levels of micronutrients like sodium and phosphorus had a huge effect on which. So if you go across the landscape, a huge effect on the distribution of the different ant species, especially mimosae, and also even the turnover rate of the ants in the trees. So, so there really are very strong knock-on effects that you might they're not immediately apparent when you first start studying a system. You're so taken with the interaction right before you of the ant and the plant that it just seems surprising that, in fact, the nutrients in the soil are having this, really profound effect that you wouldn't see unless you went out and measured it. So there, I'm sorry, I've gone slightly off topic there, except that it's related to things like biological warfare.
Yeah, very cool. Jay?
And I'll just add, just when you think about all of this coevolution and co-speciation and adaptation, it's just fun to think about these hosts over a huge range having different responses to the parasites or their other partners depending on the environment. So there's so many indirect interactions going on. There's so many opportunities for like new strategy, because all these hosts they're fighting back. They're not just passive, they're evolving at the same time. It's just wonderful to think across this huge adaptive landscape, the opportunities for all kinds of co-speciation.
Absolutely. And I'm going to throw another question to you, Jay.
Sure.
So parasitism has evolved in so many different plants, including even one lineage of gymnosperms. I'm wondering if you could sort of talk a little bit about the sort of evolutionary advantages of parasitism.
Right. So when you think about parasitism and you open up a parasitology book, it's an animal book, right? I mean, and because I think it's like 40% of animals are parasites in some way, but in plants, it's really 1%, 4,000 plants. But so it's obviously not as advantageous for plants for some reason. But when you consider like this model, like this group of parasitic plants, one advantage for being hypogeous or underground, or even being an endophyte, is you're hiding from all the herbivores. You're like, and I have some data from a study I didn't talk about on the pollination biology, where I had 140 open pollinated flowers that I was following, and 70% of those were just bitten by an animal. Like they just bit it and killed it, right? And so being underground in this type of environment is where you want to be, right?
That's awesome. And follow-up question from the audience for you, Jay. What percentage of the host species are parasitized, and how big as a negative effect does the parasite cause?
That is a great question, and something we struggled with trying to figure out in the field, because it was so hard to find the parasites and you had to dig, right? So I mean, I don't have a good answer for that. Like how many of them were parasitized? I mean, I'll guess though, but 10, 20% of the hosts were probably parasitized, but you didn't typically see the parasite really stressing out the hosts because, as I mentioned earlier, they're so water conservative, except when they're flowering. They're not losing any water really at all. And so contrast that to mistletoes that are just like pouring water out of their leaves just to draw water from their hosts. These things are underground, and I'm there. Of course, there's a negative effect on the host when you have this massive rhizome system and you're taking all this carbon. But if you wandered through the feld and you looked at dead hosts, you didn't always find parasites. Right? It's just a tough place to live. Great,
thanks. So in Jay's system, you've got an obligate parasite there, right? It's not going to live anywhere else outside of that host, except for as seeds, germinating seeds. I'm wondering if Naomi and Posy could sort of, just kind of touch on any sort of obligate associations in their systems and things that maybe, really don't live outside of those systems, or maybe those that just really flourish within the systems that you studied.
Well, I'm very interested in the phytoecious ants because they're effectively obligate symbionts of the plants. Since they're not free living, you don't find them in nests free living. We can rear them in the lab on their own, but they really live in the plants. And I mean, it's interesting because these plants have effectively given up some portion of their immune system to the ants, or the ants, they've taken over that defense function, and maybe by cultivating the fungus as well, who knows, or the fungus and the bacteria. That's what makes it too complicated. But because that habit has evolved so many times, and you have 100 different phytoecious ants, there's a lot of power to that convergence because you can look across lineages, and especially with some genomic sequencing, you might be able to identify pathways that are systematically gained or lost in the host or the obligate symbiont, and I think that will tell us a lot about these interactions, and maybe quite a lot about plant defense systems in general. So that is something that really interests me.
Yeah, absolutely. Thank you. Posy, do you have any thoughts on this one?
Yeah. So there certainly are obligate fungal symbionts in leaves. The rust pathogen, for example, is obligate, and there are examples of endophytes that are obligate symbionts. In particular, clavicipitaceous endophytes of grasses, for example, that are vertically transmitted, are obligate symbionts. But the group of endophytes that that I'm really focusing on and talked about today are not. These are endophytes that are in the environment. These are fungi that are in the environment. They infect leaves, and then they can also infect all sorts of other plants. They're quite opportunistic, overwinter in the leaf litter, and so it's a little bit, I would say, for these non-obligate symbionts, it's a little trickier, obviously, to study the the evolutionary relationship between the plant and the symbionts. And so I think that currently the work that we're doing now is really just trying to establish whether there's a sort of a genetic basis to these phenotypes, and then going forward we can start to ask more specific questions about the the possibility about an evolutionary advantage of hosting these organisms.
Great, thanks so much. And kind of a follow-up question for you, Posy, from the audience is, are there between leaf differences in the fungal communities that you have found?
Yeah, sort of getting at the same sort of point about how these are not systemic, these are not vertically transmitted or systemic fungi. So they infect leaves from the environment. And so on any individual plant, leaf to leaf, you can have a slightly different community.
That's awesome. So just kind of, many of you have alluded to this, but kind of a general question for all of you. And I think something that I hear really commonly from students and people that I talk to about, symbionts and other organisms that live in close collaboration is how much genetic material is exchanged between the two or more organisms that are living in this, this close contact. And I wonder, in your respective systems, have you documented, or has genetic exchange been documented in these systems? And could you speak to that a little bit?
I can jump in because it's easy for parasitic plants. And most people are aware of horizontal gene transfer and the wonderful stories of Rafflesia, the largest flower in the world. And the whole genome sequencing has shown that I think it's something like 1 or 2% of its whole genome is from its hosts, which is incredible. And what I love about those papers is that it raises the possibility that it's not just random transfers of genes. Like, these parasites are could be using the genes as genetic camouflage to like, so the parasite doesn't detect the host. I mean, just amazing. I mean, the host doesn't detect the parasite, but you see what I mean. But so it's well documented, and as more whole genome sequencing is going on, we're just going to find more and more of this incorporation of transgenes.
Very cool. Naomi, you want to
I
address
think it's
that?
true for insect-plant interactions that maybe you're more likely to see it in the antagonistic ones. I'm unaware of anyone who's looked at this with, or who's found this in ant plant systems, but I was just reading about this, how mealybugs manage to feed on so many different plant groups by co-opting genes from the plants themselves to detoxify the plants. It's very, very clever artistry. So I'm guessing that there'll be examples, but I don't know of it in an ant plant system.
Yeah, I think I can't think of many examples either. Although it's beginning to be studied a little bit more. But with eukaryotes, it's not as common, not as well studied. But I do recall certainly gene transfer between different leaf fungi to fungi, and endophyte to plant. So I can recall 1 or 2 examples, but I think this is kind of an exciting new area to think of what types of exchanges have happened and how common it is.
Great, thank you. I want to thank you all so much. We're going to have our final question here for the panel, and that is sort of in the spirit of a kind of a 30-second elevator pitch. What is the most fascinating aspect of your research, or the thing that you really want everyone to know, kind of the takeaway from today? And let's start with Jay.
Sure, thanks. So just the takeaway I want everyone to know is that natural history and taxonomy is amazing, and that even in a small genus like Hydenora, there's so much that's not known. I mean, I showed you the list of like a dozen synonyms. I mean, there are real species there in East Africa and the Horn of Africa, and that we need students and scientists to continue studying biodiversity just for its own sake, because it's amazing.
Awesome, thank you. Couldn't agree more. Naomi. Actually, it looks like Naomi might be frozen, so we're gonna go to Posy.
Yeah, so I guess I would say that I think the takeaway today for me is just to point out that plants defend themselves against pathogens in all sorts of different ways, and including by hosting these horizontally transmitted.
Really want to assess costs and benefits. I guess that's part of it. And in terms of a takeaway, where things are going, I just mentioned that I was interested in whole genome sequencing of ants and plants because of the power of being able to look comparatively across taxa to see how they've shaped each other. And so that to me is also a very exciting direction to look at, especially once you realize how careful, highly tuned they are to each other's signals, to reading each other's signals. There's good mutualists and sort of, or reciprocal parasites. I don't know how you view them. There's a sort of cynical view of mutualism, is that they're both tagging at self-interest. But I think to really understand cooperation, evolutionary cooperation, you have to understand that kind of self-interest in what the underlying currency is. So.
Thanks, Naomi. I'm going to go back to you, Posy, to finish your thought, or you can start over. Feel free to start over your thought.
Yeah, so I was saying that I think the takeaway is really that plants resist pathogens and defend themselves in a variety of different ways, and including by hosting these horizontally transmitted fungal leaf endophytes. The one thing that's just really kind of cool and interesting about the symbiosis, it's really like a community symbiosis, right? So endophytes exist in these complex communities with individuals, doing different things and modifying disease severity in different ways. And so I would say that, trying to understand the consequence of this community for the host just involves, kind of painstakingly detailed work, understanding the assembly and the composition of the community. And one thing though that's exciting about what we're finding is that, we are seeing evidence that the host can exert control over the assembly of the community. And so, one exciting big next question is really trying to better understand which host traits and genes underlie these effects on composition and on function. And working in the populous system is a great place to ask this question because of these genomic resources that are available. So right now we're in the middle of genome-wide association studies, using the SNP data to try to identify genes that are associated with the microbiome phenotypes that we're working with. In addition, we can also do sort of plant gene expression experiments in controlled settings where we inoculate the plant with individual endophytes and communities of endophytes to evaluate how the endophytes are modulating the host response to endophyte colonization, but then also to the pathogen. So I think kind of trying to work out the sort of genetic influence on endophyte assembly, colonization and assembly, I think is going to be an important piece in terms of understanding sort of the evolutionary basis of this relationship, but then also moving forward if we want to try to apply some of these results to agriculture.
This has been really wonderful. Enjoyed talking with all of you. You gave some great talks today, which were super engaging. I want to thank you so much for participating in this symposium. And to our audience, we're going to do this again tomorrow with 3 more speakers. And if you have not registered if you didn't register twice, then you're only registered for today. So check out the link that we've got in the chat there, and be sure to register for tomorrow and tune in again. We'll be talking more about symbiosis. Thanks so much, and I hope you all have a great night.
Thanks, Susan.
Thank you.
Thanks, Jay and Naomi.
Yeah, it was fun. See you tomorrow.
Bye.
Sounds good.
Bye.
Thanks.